Assalamualaikum. Bismillahirrahmanirrahim. Assalamualaikum and good day, ladies and gentlemen. I'm Hazima Mozam and I welcome you all to a public lecture on microbial biofilm formation and its importance as a drug target. This lecture will be delivered by Professor Dr. Ute Römling. We are very grateful to Professor Dr. Ute Römling for her time and for kind visit to Comstech. We will begin today with the recitation from Holy Quran.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم الرحمن علم القرآن خلق الإنسان علمه البيان الشمس والقمر بحسبان والنجم والشجر يسجدان والسماء رفعها ووضع الميزان ألا تطغوا في الميزان وأقيموا الوزن بالقسط ولا تخسروا الميزان صدق الله العظيم.
Jazakallah. With this, now I would request Coordinator General Comstech, Professor Dr. Muhammad Iqbal Chaudhry, to welcome and introduce our guest speaker for today.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم. Assalamu alaikum and good afternoon, good morning and good evening, ladies and gentlemen. I'm extremely pleased to introduce someone I know since many years and have been always fascinated from the talent and the spirit of Professor Dr. Ute Römling. A very renowned scientist. She's also a Comstech distinguished scholar and also an adjunct faculty at the International Center for Chemical and Biological Sciences. Professor Ute is very well known in the field of biomedical sciences, particularly in the field of infectious diseases. She is currently working as a full professor in Karolinska Institute. She's a professor of medical microbiology in Stockholm, Sweden. Her research focuses on the molecular mechanisms of biofilm formation. As you know, biofilm formation is an extremely important mechanism for which is involved in antibiotic resistance. And her research has been largely targeted towards enterobacteria such as Salmonella typhium and persistent mechanism of Pseudomonas aeruginosa, worldwide spread clone C. Professor Römling has over 130 publications of her original work, 41 peer-reviewed reviews, 2 peer-reviewed protocols, 10 popular scientific publications, 15 book chapters, and 1 book editorial. In addition, she has received research grants from Swedish Research Council, ALF Foundation, European Commission, and German Research Foundation. In 2000, she received the Young Investigator Award for research in clinical microbiology and Infectious Diseases 2000 of the ESMID, European Society of Clinical Microbiology and Infectious Diseases. Furthermore, Professor Römling has been an associate faculty at the International Center for Chemical and Biological Sciences in University of Karachi, Pakistan, where she has been supervising a very large number of young scholars in many interesting projects. She's a member of the European Academy of Microbiology since 2017 and member of USERN since 2020. Professor Römling, apart from her wonderful educational credential and her the impact of her research in the field of microbiology, is a wonderful human being. She thinks globally and she thinks of benefiting lots of young people. She traveled all the way from Stockholm to Karachi and then to Islamabad only with the objective of developing capacity and helping young people in their research. And I think this is something which is extremely important, something which we all cherish. With this, I would like to invite Professor Dr. Ute Römling for her presentation. A bit late, but anyway. Focus कीजिए.
First of all, I would like to thank Comstech, the president, Dr. Arif Alvi, and especially Professor Iqbal Chowdhury and his entire team at Comstech for inviting me. It's a big honor to be here. And I would like to present today some of our work on microbial biofilm formation and also on the project that we have conducted together with Professor Chowdhury and his team on its importance as a drug target. What are biofilms? Maybe many of you already know what are biofilms, but I would like to give a very broad introduction into biofilm formation. Biofilms are actually matrix-enclosed bacterial populations adherent to each other and or to surfaces or interfaces. And this very broad definition of biofilm formation comes from the father of biofilm research, Bill Costerton. What does this definition mean? It means basically that any aggregation already of, for example, 2 autonomous bacterial cells can be considered as a beginning of biofilm formation. So why do we focus on biofilms? Some time ago, actually, the National Institutes of Health have estimated that approximately 60 to 80% of the human bacterial infections are caused by biofilm-forming bacteria. And why is this number that huge? Biofilms actually are tolerant against antibiotics. Biofilms are also tolerant against the immune response, like antibodies and phagocytosis through macrophages, which leads to frustrated phagocytosis and tissue damage rather than to eradication of the organism. Modern medicine uses indwelling devices, and these are prone to biofilm infections. Thus, biofilm infections will accompany us certainly for a longer time. In addition, the population becomes older and requires more medical care. Also, this makes the individual prone to biofilm infections. Our model of biofilm formation is based on an agar plate grown biofilm of Salmonella typhimurium that we called the RADA morphotype. And RADA stands for red, dry, and rough. And I think you can appreciate on the left side, we have a colony morphology of Salmonella typhimurium that at this body temperature of 37 degrees does not form biofilms. While on the right side, you see a colony of Salmonella typhimurium that forms biofilms. And the dye is taken up by extracellular matrix components produced by the biofilm forming microbes. We have identified also the major regulator of biofilm formation that is called CSGD. Upon expression of CSGD, we see this extensive biofilm formation, the multicellular structure in liquid culture, and also biofilm formation at the wall of an abiotic glass, for example, surface. This type of biofilm often serves as a model to investigate biofilm formation. On the other hand, when CSGD, the major biofilm regulator under these conditions, is deleted, we see a white colony and we don't see any multicellular behavior, neither in liquid culture, where we only have singular planktonic cells, and also we don't see adherence to the wall of the well of this abiotic glass surface. However, is this biofilm model that we see on an agar plate in the laboratory also relevant in reality? And if so, where does this occur? We know today that this type of biofilm that is described originally on an agar plate is widely applicable also in a lot of infectious disease conditions. For example, this type of biofilm occurs on a number of abiotic surfaces. It occurs in the environment as these biofilms are spore-like structures that can resist desiccation for months. It is also found on epithelial cells. It is found on plant surface, in interaction with fungi, in cancer tissue, and on gallstone. And the impact of this biofilm formation of Salmonella typhimurium is equally wide. This biofilm makes the cells tolerant against disinfectant. They are resistant against desiccation. They colonize and persist in different tissues. They also cause a chronic carrier status, for example, on gallstones. They promote partially internalization into host cells, and they induce the immune system. And they are responsible for transmission of the organism from individual to individual. In addition, almost 50% of Salmonella typhimurium outbreaks are caused by fresh produce, such as tomatoes, spinach, and melons, where also biofilm formation plays a major role. On this basis, we have some time ago started a project with Professor Chowdhury on the identification of antibiofilm compounds against the gastrointestinal pathogen Salmonella typhimurium that was recently published in the journal Infection and Immunity. During the screen for antibiofilm compound, we realized that macrolides have an impact on biofilm formation. And the macrolide azithromycin, as shown here, is not a antibiofilm compound, as the growth inhibition occurs at lower concentration than inhibition of biofilm formation. While on the other hand, the macrolide clarithromycin is an antibiofilm compound, as growth inhibition occurs much later as biofilm inhibition. We can see upon application of subinhibitory concentrations of the macrolide clarithromycin, that the cells aggregates gradually dissolve and the biofilm is actually disappears until at the end at subinhibitory concentrations, we have only single planktonic cells. We also can see a quite distinct response of the bacteria when subinhibitory concentrations of clarithromycin are applied. Among the 4,800 Salmonella typhimurium genes, over 350 genes show at least fourfold divergent expression upon exposure to clarithromycin. This very specific response indicates that the cell tries to defend itself against the antibiofilm compound. To summarize the response of Salmonella typhimurium against the subinhibitory concentrations of clarithromycin, we could see that we have, as we would have expected also, a distinct reduction of genes involved in biofilm formation. And also what we also would have expected, we see an upregulation actually of the antimicrobe, of the targets of the antibiotics, and these are the ribosomes. But in addition, we see a much broader physiological response to the antibiotic, as we see a upregulated heat shock response. We see also a shift to oxygen and energy depleted status, indicating also that we have in this case reactive oxygen species are actually not involved in the response to this type of antibiotic. We see a promotion of ethanolamine, propan-1,2-diol, and L-arginine as a carbon source, and we see a downregulation of the Krebs cycle and other metabolism that channel into the oxidative phosphorylation consistent with the oxygen depletion status. Biofilm formation is highly regulated by a lot of different conditions and by a lot of different components that are encoded on the genome. And a major regulator of biofilm formation is actually second messenger signaling. Second messenger signaling has been discovered by Earl Wilbur Sutherland in 1967. He observed that the hormone epinephrine stimulates the liver to convert glycogen to glucose. And this conversion required a soluble intracellular molecule that has been identified as cyclic AMP. So second messenger signals are produced in response to a first signal. And those are usually small molecules that are rapidly synthesized or degraded by enzymes and or also released by an ion channel. And those second messenger enable a highly flexible response, a highly flexible spatial and temporal regulation of the signal for the cells to highly flexibly respond. These second messenger signals can be of different nature. For example, hydrophobic membrane standing molecules like phosphatidylinositol, water-soluble molecules like cyclic AMP, as Earl Sutherland discovered, but also other nucleotides like cyclic GMP, cyclic di-GMP, which I will come back to, and other cyclic dinucleotides. But it can also be gases like CO and nitric oxide. A second messenger signaling to signaling cascade starts always with a first messenger. So the cell integrates several of these signals. And for example, a first messenger can be light and amino acids, as diverse as these different compounds or physical parameters. On the other hand, there can also be intracellular first messengers, like the nutrient and the redox status and or the temperature. But these first messengers are not restricted to those signals. There can be a high diversity and multitude of these signals that the cells senses by different components. To exemplify the signaling cascade of a second messenger signaling system, I would like to do that using the ubiquitous second messenger cyclic di-GMP as an example. So upon reception of the first signal, proteins like the GTDF domains which synthesizes the first messengers are activated and synthesize cyclic di-GMP. Cyclic di-GMP as a second messenger might diffuse only locally or might diffuse globally in order to meet a receptor, in this case, pill-set receptor. Receptors itself can have effector functions and lead to a target response. And on the other hand, receptors can also work through protein interactions to successively cause a target response. The signal is subsequently then shut off by phosphodiesterase activity. That phosphodiesterase hydrolyze the signal, and those phosphodiesterases, in the case of cyclic di-GMP signaling, are so-called ERL domain proteins. ERL domain proteins significantly contribute to the spatial and temporal regulation of the signal and are also regulated heavily by so basically first messengers that regulate the activity of phosphodiesterases. Through such a second messenger signaling system, a physiological response in the millisecond to hours range can be exerted. Not only spatial but highly temporally broad is a response by second messenger signaling. The common view on prokaryotic signaling system has actually been imprinted by research on cyclic AMP in Escherichia coli. And cyclic AMP in Escherichia coli exerts quite a linear signaling cascade. In the absence of glucose, the adenylate cyclase is phosphorylated by the phosphotransfer system in the inner membrane. Activated adenylate cyclase then produces cyclic AMP, and cyclic AMP is binding to the cyclic AMP receptor protein, which regulates hundreds of genes on the Escherichia coli chromosome. We have also a cyclic AMP phosphodiesterase, which degrades the system, and cyclic AMP can also be secreted, but these two components usually do not play a role in this signaling system. The discovery of cyclic di-GMP came to a surprise, because also Stuart Schreiber states life would not exist with macromolecules alone. The bacteria were for a long time considered to be too small to require complex secondary messenger signaling system. So the focus was predominantly on secreted small molecules and also on the quorum sensing, which is an extracellular signaling system. Upon the rediscovery of cyclic di-GMP by us and other groups in 2004, we have actually defined a role for cyclic di-GMP as an ubiquitous second messenger in bacteria. And not only we have identified cyclic di-GMPs as a ubiquitous second messenger, we could also show that upon high levels of cyclic di-GMP, we promote sessility of single cells, which leads to biofilm formation and to the production of adhesive extracellular matrix components. On the other hand, when we have low cyclic di-GMP, promoted by the activity of the ERL phosphodiesterase, we promote a cellular status that responds to motility. So we see high motility upon low cyclic diet GMP levels. This switch from motility to sessility is actually a basic lifestyle transition of a single bacterial cell. And we and others have shown that this basic lifestyle regulation by cyclic diet GMP is valid not only in Salmonella typhimurium and closely related bacteria, but in any other bacterial species that has been investigated. This shows the developmental cycle of a biofilm starting from single planktonic cells to a mature biofilm where single cells can again detach to start this life cycle again. And cyclic diet GMP signaling plays basically a role on any of these stages in the development of biofilm formation from single planktonic cells sensing a surface to the mature biofilm and detachment of planktonic cells again. In addition, cyclic diet GMP also contributes to very many different physiological features of this biofilm, for example, distinctively also to antibiotic tolerance. Besides the basic lifestyle transition between sessility and motility, we and others could also show that cyclic diet GMP regulates the transition between biofilm formation and acute virulence. So upon high cyclic diet GMP levels, again, sessility is promoted, which leads to chronic infection status, basically to a chronic virulence, and for example, also to commensalisms like a gut colonization. On the other hand, when we have low cyclic diet GMP levels, acute virulence is promoted. Acute virulence is defined by, for example, excessive stimulation of a pro-inflammatory cytokine response and by virulence properties like, for example, invasion of host cells. A hallmark of the cyclic diet GMP signaling system, and what came to a very surprise of when this signaling system was first discovered, is the abundance of cyclic diet GMP turnover proteins encoded by one bacterial genome. And that has been designated as a cyclic diet GMP IQ. What we can say is that, or what we observe is that within the phyllum, there is a linear relationship basically between genome size and the number of cyclic diet GMP turnover proteins. However, the slope of this curve can be differentially steep, and especially actually gamma proteobacteria produce a high number of cyclic diet GMP turnover proteins, while actinobacteria, as you can see, have a much, the slope is much lower, and we have much lower number of cyclic diet GMP turnover proteins. Upon reduction of genome size due to host adaptation, the cyclic diet GMP signaling system usually disappears. In one of our model organisms, Escherichia coli, the cyclic diet GMP signaling system is of moderate complexity. We have in total 31 or 32, dependent how you count it, cyclic diet GMP turnover proteins that either synthesize or degrade the second messenger, and in some cases can also bind this molecule. However, what happens when a novel second messenger signaling system comes in? We have observed at some point that the right border high pathogenicity island of a commensal strain contains a novel signaling module. And this novel signaling module actually contains also a homologue of the dinucleotide cyclase DNCV that has been found previously in Vibrio cholerae altor. And DNCV produces a novel second messenger that is called cyclic AMP-GMP. Upstream of DMCV, there is a protein encoded that is called CAPV. This is a patatin-like phospholipase, and this patatin-like phospholipase has been designated as a receptor for the second messenger cyclic AMP-GMP. We could show that cyclic AMP-GMP is actually also distinctively involved in the regulation of biofilm formation and motility in Escherichia coli. However, in the case of cyclic AMP-GMP, this molecule actually inhibits biofilm formation instead of promoting it. We have also purified DNCV and shown that in vitro cyclic AMP-GMP is produced, and in addition, a number of minor subproducts are observed, so other cyclic dinucleotides. This shows that DNCV is actually a much more promiscuous protein than, for example, the GGDF digranulate cyclases, which predominantly but not exclusively produce cyclic diGMP. So when we observe a novel second messenger signaling system, we would usually ask a number of obvious questions, and those questions are: what are the mechanisms of cyclic AMP-GMP to downregulate the target output, in this case motility and biofilm formation? What is the identity of the receptor? Which enzymes degrade cyclic AMP-GMP? And are there alternative targets beside the biofilm regulator, CSGD, and motility? And we're going to answer basically these first two questions. And on the way to answer these first two questions, actually, we changed our direction to some extent. And I will tell you a little story about scientific serendipity, which we found worth enough to proceed. So this first question basically then channeled into the question, or into the statement of discovery of evolution of a patatin-like phospholipase. So we're asking the questions: what are the mechanisms of cyclic AMP-GMP to downregulate motility and biofilm formation? And this resulted in the discovery that the cyclic AMP-GMP receptor variant CAPV Q329R adapts new functionalities. So how did we discover it? We first expressed the patatin-like phospholipase CAPV and assessed its effect on motility. When we did this, we did not see any effect. However, when we assessed the effect of a variant that has been derived from some uncloning error, we could see that this variant significantly downregulated motility. And not only did it downregulate motility, it caused also an extensive filamentation of the E. coli cells. Filamentation usually occurs upon exposure to antibiotics and or to substantial stresses, and indicates that cell division in bacteria is inhibited. So cell division usually occurs upon the replication of the chromosome. Then we have the elongation of the bacterial cell and the nucleoid segregation. Upon sufficient segregation, the FTSZ ring formation occurs, and invagination in order to divide the cells into two daughter cells. And we were wondering at which level the new variant CAPV Q329R affects cell division. When we look at the filaments under the microscope, we can see that we still have FTSZ ring formation here in green, but FTSZ rings are much more scarce than in the and occur at at longer distances in between than in the wild type cells. We could also see that the nucleoid formation and nucleoid separation is partially inhibited, and that invagination is hardly visible. So most likely the block in cell division occurs downstream of FDSZ ring formation, but might also partially occur upstream of FDSZ ring formation. We were then wondering, as we have observed this effect in E. coli laboratory strain, whether this effect occurs also is wide broader and occurs in other E. coli strains and probably also in other bacterial species. In order to answer this question, we expressed the variant CAP-V-Q129R in a number of commensal E. coli strains, and also in urinary tract infection isolates, and we could see that in most of the strains, the expression of this patatin-like phospholipase variant causes substantial cell filamentation. In order to investigate the phenotype of biofilm formation, we could also see that in all of these bacteria, biofilm formation is differentially regulated by the overexpression of the variant. And in most of the cases, biofilm formation, which occurs here at body temperature, is downregulated by expression of the patatin-like phospholipase variant CAP-V-Q329R. The DNCV, the dinucleotide cyclase DNCV, and its associated genes are actually have been demonstrated to be part of a phage defense system and to restrict the number of and to restrict phage infection. So we were wondering whether our variant is involved in the transition or in the mediation of susceptibility to a phage, and we choose phage P1, a common phage that infects Escherichia coli. In order to, when we looked at the phage susceptibility upon overexpression of the CAP-V variant, we could see that in contrast to what has been reported in the literature, this variant enhances the susceptibility to phage P1. This indicates that we have a substantial rearrangement of the functionality of a phospholipase in just by the substitution of one amino acid. Patatin-like phospholipases have been scarcely investigated in bacteria. And when they have been investigated in their functionality as virulence factors. Usually those proteins are produced by the bacterial cells, but they are not active. They are activated upon injection into host cells by host factors. For example, XOU can be activated by monoubiquination or by polyubiquination. And these phospholipases can also have not only lysis cells, but they can also exert signaling functions. And usually they are belonging to a subclass of phospholipases type A2, which lyses the 2 as an ester bond. So in summary, CAP-V is a patatin-like phospholipase. It is, and in this case, it's a member of this family. And these enzymes actually occur in all domains of life. And they have not only functions as cytotoxic agents, they also function as signaling, as in a second messenger signaling cascade, or in the transition of fatty acids. Patatin-like phospholipase variants are associated with human diseases like non-alcoholic fatty liver disease. But in bacteria, patatin-like phospholipases can be virulence factors, but the physiological function of these patatin-like phospholipases have been very poorly investigated. Our variant that we have discovered is an example of a rapid evolution of protein functionality, but this variant does not occur naturally in isolates. The molecular mechanisms of the various physiological and metabolic changes caused by CAPV overexpression still need to be unraveled. I will then shortly present evolutionary aspects of cyclic diet GMP signaling. And we were asking the questions, why is cyclic diet GMP signaling so flexible in evolution? As we have observed that in some bacterial species, cyclic diet GMP signaling can rapidly disappear. So we were systematically screening different bacterial genomes for the presence and absence of cyclic diet GMP signaling. And we could see that the disappearance of the cyclic diet GMP network from a genus of gamma proteobacteria and acquisition of cyclic diet GMP signaling network by streptococcal species. And this is shown here. On the left side, there is a phylogenetic tree of the gamma proteobacteria, and most families in this phylum, they contain members that have an abundance of the cyclic diet GMP signaling system. However, the family Morganellaceae, actually its members do not contain cyclic diet GMP signaling besides Proteus species, which had maintained one diagonalate cyclase. On the other hand, streptococcal species, like the pathogenic Streptococcus pneumoniae and Streptococcus pyogenes, they lack the cyclic diet GMP signaling system. However, in a number of streptococcal species, we can observe one diagonalate cyclase. Upon the evolution of a cyclic diet GMP signaling system, usually upon reduction of this system, reduction is associated with a subsequent lack of functionality. And that has been demonstrated in Staphylococcus aureus, which actually has one apparent diagonalate cyclase, which, however, has not proven to be functional. Also, bioinformatic analysis would have predicted functionality. So the production of poly-N-acetylglucosamine, that is cyclic diet GMP signaling regulated in, for example, Escherichia coli, is also regulated by the GTDF domain protein in Staphylococcus aureus. However, the catalytic activity of the GTDF domain protein is not involved. On this background, we were wondering are the GTDF domain proteins indeed of Proteus and Staphylococcus and Streptococcus indeed diagonalate cyclases? Looking at the bioinformatically on this protein, it looks like they could be functional. In Proteus species when compared to experimentally demonstrated functional proteins. We then conducted experiments in order to show functionality. One experiment is based on riboswitches, so-called off-riboswitches, which upon sensing of the cyclic di-GMP switch off the downstream expression of a gene. In this case, a beta-galactosidase, which is actually causing then the catalytic, excuse me, the catalytic turnover of a substrate into a dye. We could show that when we overexpress the digranulocyte, the GTDF domain protein from Proteus, we can see that we get a downregulation of the catalytic activity and indicating that we have high cyclic di-GMP produced and no cyclic di-GMP produced and no substrate is made. When we, however, overexpress a catalytic mutant of the GTDF domain protein, we see no change in catalytic activity, indicating that indeed we have a digranulate cyclase activity. We confirmed the digranulate cyclase activity by additional means. We also measured cyclic di-GMP levels in cell extract, and we could show that not only the GTDF domain protein from Proteus mirabilis, also the GTDF domain protein from Streptococcus are indeed digranulate cyclases, and upon overexpression of a catalytically inactive mutant, we see no expression of the substrate. We could also predict the signaling pathways for these digranulate cyclases, which produces cyclic di-GMP, as the digranulate cyclases is encoded actually in the context of a operon that is encoding for enzymes involved in the synthesis of cellulose. Cellulose is an abundant macromolecule that is found in the biofilm matrix, in the matrix of biofilms. However, Proteus species were not known before to produce cellulose as a biofilm matrix. So in conclusion, we can say that in the family of Morganelaceae, all investigated Proteus species, including the major pathogen Proteus mirabilis, encodes a pus GTDF domain protein. And this GTDF domain protein is found in the context of a cellulose biosynthesis operon. Proteus mirabilis harbors two cellulose biosynthesis operon, and we can at the moment not experimentally verify the function of the pus GTDF domain protein in this context, as we could not detect cellulose biosynthesis and motility to be affected upon overexpression. However, we might indicate other environmental conditions in order to elucidate this question. On the other hand, we did basically a similar analysis for the streptococcal GTDF domain protein, and we could indicate by bioinformatic analysis, yes, these proteins might also be functional. They contain all the consensus motifs required for catalytic activity. And again, to our surprise, we could find the digranulate cyclase in the context of what we have identified as a gene operon required for the biosynthesis of a cellulose or a cellulose-like exopolysaccharide. So in conclusion, we can say that the streptococcal species of the bovis, salivarius, and pyogenes, and suis and Henry clades harbor a GTDF domain protein, and in some cases even two. The GTDF domain proteins are discriminated by highly divergent amino acid sequences with a common partially variable domain structure of transmembrane pass GTDF. Most GTDF domain proteins are part of a horizontally transferred cellulose-like biosynthesis operon. And the GTDF domain proteins are involved in a novel type of biofilm formation that still needs to be demonstrated by promoting the production of cellulose or a cellulose-like exopolysaccharide. What can be the practical implication of this finding? The practical implications of this finding can be twofold. On the one hand, cyclic diGMP signaling and cellulose biosynthesis might be a target for antibiofilm therapy. For example, Streptococcus gallolyticus is an occasional pathogen involved in triggering cancer. On the other hand, cyclic diGMP signaling and cellulose biosynthesis might also be a target for probiotic therapy, as promotion of biofilm formation in a probiotic bacterium, Streptococcus salivarius, might enhance its beneficial probiotic properties. And I think I have to conclude my lecture, and then I would just like to thank all the people involved in this work, especially what I presented in more detail was performed by Fenyang Li, Ying Liu, Changhan Li, Sulman Shafiq, and Munira Safar. We had a very productive collaboration and is still ongoing with Professor Iqbal Chowdhury, Humaira Yahan, and all his co-workers. And without all the other collaborators that we had the pleasure to work with, this work would not have been possible. Thank you very much for your attention.
Thank you very much, Professor Dr. Römling, for a very interesting and comprehensive lecture. With your permission, I would request our in-person and virtual participants for the question and answers. We can begin with the questions from the in-person participants. Do we have any questions here? Okay.
Professor Römling, very interesting and very informative presentation. Professor, you know that antimicrobial resistance is a global threat. Can these anti-biofilm compounds can be used to reduce the antimicrobial resistance? Number one. Number two is any probiotic or any drug which is available in certain using these compounds.
Biofilm formation basically is a cause for chronic infection. And some biofilm associated diseases we can only eradicate when we remove the cause, for example. That is a case, for example, by catheter-associated urinary tract infections. In these cases, it is advisable to remove the catheter instead of treating the biofilm that is formed on the catheter surface. This said is that definitely biofilm, and on the other hand, there is also known that some of the some antimicrobial substances already in clinical use are more effective against biofilms than other substances. However, with our research, we would like to extend maybe this knowledge base and to find new and perhaps or hopefully more effective combinations also of drugs which can effectively combat biofilms, biofilm-based infections.
Thank you.
Thank you very much, madam. Do we have another question here?
Yes. Thank you so much, Dr. Römling, for a very informative talk. My question is very simple. I'm just wondering, how will you differentiate between planktonic or free living form and biofilm forming pathogen? Because in one of your slides, you mentioned that after treatment, biofilm was removed and you just got few of the planktonic cells. So how in vitro, in lab condition, we easily differentiate between free-forming and biofilm forming pathogens? Thank you.
Well, it is said that, for example, almost any microbe, autonomous microbe, can form a biofilm. But usually how we define free-living planktonic cells is that we see single cells. For example, under the microscope, or we see no attachment of cells to a abiotic or to a biotic surface. However, also of course, in the growth medium, practically speaking, you can see cell aggregates. And we define these cell aggregates already as a type of biofilm. These cell aggregates might be visible by the naked eye, or they can be visualized under the microscope.
Thank you very much, madam.
Thank you.
Do we have any other question here? Okay, so we have some questions in the chat box, madam, for you. There's this one question from Mr. Zahidullah. He's asking, although there are many synthetic chemicals for the removal of biofilm, is there any biological way of mitigation for the biofilm?
Biological, yes. There are, for example, different bacteria affect biofilm formation or different microbes can affect biofilm formation of other microbes. So, for example, some microbes produce exopolysaccharides that prevent adhesion to a biotic or biotic surface. On the other hand, the first quorum sensing inhibitors, the furanones, they have also been found produced by algae, basically by a red algae. So they are natural compounds, but they are definitely, in spite of that they're natural, they are very toxic to a metazoan organism. On the other hand, also when we look at the human body, we produce a lot of anti-biofilm compounds, and maybe the most well-investigated one is the antimicrobial peptide LL37, which does not only have an antimicrobial effect, but it also has actually at sub-inhibitory concentration a much more efficient anti-biofilm effect.
Thank you very much, madam. So we have another question from Ms. Zeba Saeed. She's asking, is the composition of biofilm matrix associated with the severity of infection?
I'm not aware that of such studies.
There is another question from Mr. Shazeb. He's asking how cyclic di-GMP is different from cyclic AMP and how cyclic di-GMP is produced.
So cyclic AMP is basically cyclic AMP is basically one molecule of AMP that is cyclase by phosphate D-ester bond, while cyclic di-GMP consists of two molecules of GMP that form basically a cycle, a 3' bonded. So this makes this molecule much larger. And what was the other question?
How is it produced?
How it is produced. Those molecules are distinctively produced by distinct enzymes. In the case of cyclic di-GMP, those enzymes are characterized by a so-called GGDF domain. This domain contains a GGDF motif, and these domains can basically relatively readily identified in bacterial genomes. The cyclic AMP is produced by a adenylate cyclase, which is structurally similar to the digonolate cyclase, but not on the amino acid level.
mainly Krebs cycle है हमारे पास तो प्रोटीन सिंथेसिस में इन्वॉल्व होते हैं
English.
Second, increase the cell content.
Further was starting a wait for my Islamic University Power and a full question from me that we are this protein synthesis there is also the influence of microorganismal growth. So Krebs cycle and the defining about the recent microorganisms and their growth regarding the antibiotics. So what's the difference between these two cycles?
Are you talking about the cycle that produce cyclic GMP and the Krebs cycle?
The Krebs cycle. A difference between two.
He's basically asking the difference between the Krebs cycle and the cycle that produces cyclic GMP. I don't know how is that associated, but.
The Krebs cycle is a metabolic pathway that starts basically when we can say maybe with a glucose as a sugar, and that is metabolized, and this and that results in a compound that is called acetyl-CoA, and the acetyl-CoA is channeled into the Krebs cycle in order to produce then carbon dioxide. So basically, this is an oxidation of the sugar in order to produce energy. This cycle is a metabolic cycle, so to say, not an enzyme, but a metabolic sequence of different enzymatic reactions.
Thank you very much, madam. Just a last question from our virtual audience. So here is Mr. Mustafa. He's asking that anti-biofilm therapy does not going to, in your opinion, anti-biofilm therapy does not going to disturb natural microbiota residing inside the organism.
Any antibiotic therapy is basically disturbing to some extent the organism and the physiology of any organism. That is why we look for more effective compounds that target specifically the biofilm in order that we need to disturb the organisms as little as possible. Just to give an example, when we have cystic fibrosis patients, which are prone to infections by Pseudomonas aeruginosa, and they, once cystic fibrosis patients are infected by Pseudomonas aeruginosa, they can colonize the lung lifelong. So for over a decade, for example, the patient consumes kilograms of antibiotics.
Thank you very much, madam. With this, we come to end of our your talk and the question answer session. With this, I would request Professor Dr. Ute Römling, coordinator for his closing remarks.
Thank you very much, Ute, for an extremely fascinating presentation, very insightful science. And we fully understand and appreciate the importance of anti-biofilm compounds. And for that, it's extremely important to identify the targets, the validated targets. I have a very personal experience of the impact of biofilm. My son gone through a surgical jaw reconstruction, and there was a small implant which was placed in his teeth, which eventually developed infection, and doctors keep treating him with tons of antibiotics. And poor boy wasn't really able to be treated with any one of these antibiotics because there was no anti-biofilm compounds was available. Till again a surgery was performed and this implant was removed. So this is extremely important for the development of anti-biofilm compound, understanding the chemistry and biology of biofilms, and as well as identifying validated targets which can be used for discovery and development are of imperative importance. So with this, I'd like to thank you for your visit to Islamabad and for this wonderful presentation. Certainly an extremely enriching and very knowledgeable. Thank you very much.
Thank you very much, sir. With this, I would request you to please present a certificate of appreciation and a comstech sheet to our guest speaker today.
Okay, so Ute, this is our shield, and this is made of walnut wood, which is a very high quality, extremely expensive, very rare wood, and only the rich and famous can afford it.
Thanks very much.
If you place it here, I'll present this to you as a token of appreciation for the presentation. Thank you. आप लोगों ने देखा इसे?
Mashallah.
With this, we come to end of our lecture today. I would like to thank all the participants who joined us virtually, and special thanks to all our in-person participants who joined us, and to all the participants who want to have recording of this lecture. This lecture is recorded and will be available on comstech's YouTube channel, so you can see it there afterwards as well. Thank you very much, and hope to see you again in another event of comstech. Bye.